So class, in this recording, we're going to focus on the cell cycle. And as we look at our cell cycle, this is how we make more cells in our bodies. It's how we divide our cells. And cell cycle can be broken up into two broad phases. We have interphase in green on the bottom and M for mitotic phase up here on the top. And this is a circular diagram representing the cyclic nature of this process. And as we're looking at interphase has three subphases: G1, G2, and then the S, or synthesis phase. G1 is for the first gap, G2 is for the second gap. And then as we look at mitotic phase can be broken up into multiple subphases where we have prophase, metaphase, anaphase, and interphase, whereas it is going to be centered around replication of the nucleus, and then cytokinesis, which is going to be focused on the replication or division of the cytoplasm. So as we look at this first gap phase, this is the interval time, or the amount of time that begins when the daughter cell is initially made and when the DNA replication begins. And during this phase, the cell is going to carry out very normal tasks. It's going to go through regular metabolic activities and accumulate additional materials for the beginning of DNA replication, which begins in S phase, or synthesis phase, where we synthesize or replicate the nuclear genome and the centrioles needed to help us with the mitotic phase. Gap 2 phase is going to begin after we've finished replicating our nuclear genome and right before we start the process of replicating the nucleus, or mitotic phase. This gap 2 phase is going to primarily focus on fixing DNA replication errors, adding more organelles, adding more mass in cytoplasm, and additional enzymes needed to help regulate the cell division process. As we look at M phase, M for mitotic phase, this is when we replicate the nucleus. And that at the very end of M phase, we pinch off the cell to form two new daughter cells, where we pinch off the cytoplasm in particular. There's a side phase called G0, or gap 0. This G0, or G0 phase, is going to describe what happens to some cells that just stop dividing. There are some cells in our bodies, like skeletal muscle cells or motor neurons, that once we reach adulthood, instead of constantly replicating, they just have a very long lifespan. They can last for years or decades. So depending on the cell type we're looking at and the age of the individual and the particular physiological environment, cell cycle duration is going to vary a lot. We can have a very short duration of less than 2 days or a very long duration of over a decade. Now when we look at mitosis, the main goal of mitosis is to help us have produce daughter cells that have two copies of the genome. And when we look at this genome, we as human beings are going to be diploid organisms, which means that we have two copies of our genome in most cells of our bodies. And so when we make new daughter cells, each daughter cell is going to also need to be diploid. We need to have two copies of our genome. During mitosis, we need to divide the nucleus in order to form two new nuclei. During mitosis, we are going to use this process of producing, or we're going to go through cellular division and mitosis so that we can grow, increase the volume and mass of all of our tissues and organs after we're born. We replace dead cells in our body, and we cause repair to tissues within our bodies. Mitosis is broken up into 4 key phases: prophase, metaphase, anaphase, and telophase. And as we look at those 4 phases, we'll dive into prophase first. And as we look at prophase is going to be primarily highlighted by taking our genetic material and condensing it into chromosomes. So the beginning of mitosis is when you go from not seeing a chromosome in the nucleus to suddenly being able to see the chromosomes within the nucleus. Condensing the genetic material into chromosomes makes it easier to spread apart or distribute the genetic material to the daughter cells. When we think of chromatin, you could think of it as kind of like a rat's nest of DNA. It's very hard to separate DNA strands when they're all unwound and intertwined with each other. We as human beings typically have 46 chromosomes in each of our cells. We have 23 different chromosomes, and we have 2 sets of 23. So 23 plus 23 gives us 46 chromosomes. And each chromosome is going to have two chromatids, so at minimum of two. So those little branches coming off of the chromosome are each branch being referred to as a chromatid. As we look at prophase, during prophase, another key event is that the nuclear envelope, that double phospholipid bilayer around the nucleus, disintegrates. And the centrioles are going to sprout spindle fibers, or very long microtubules that start growing towards those chromosomes. Some of the fibers are going to push the centrioles to opposite poles or opposite ends of the cell, and then some of those mitotic tubules or spindle fibers are going to attach to a part of the chromosome known as the kinetochore, which is going to be a protein attachment point at the center of each chromosome. And the very center of this chromosome is referred to as a centromere. So if we have a chromosome here, here's our chromosome, and then we have the centromere in the middle, and then in the centromere we have, shown in yellow here, two kinetochore proteins, and then in blue, microtubules can attach to those kinetochore proteins. As we look at metaphase, metaphase is defined as the stage of mitosis when all of those chromosomes are in a straight line or aligned along a cell equator. The spindle fibers are going to keep pushing all the chromosomes to the very middle and have connected to the kinetochores, and we are going to be able to see the centrioles as kind of a small star or aster-like structure at opposite poles of the cell. So here, as we look at this figure, we can see prophase as both a drawing here and as a photomicrograph on the right. And then here in the lower left, we have a diagram of metaphase, and here we have a photomicrograph of metaphase. After metaphase, we have anaphase, and during this phase, we're going to take the chromosomes and rip them in half. We're going to rip the two sister chromatids apart at the middle of the chromosome, or the centromere. And those single-stranded daughter chromosomes are then going to be transported or migrated to the poles of the cells as motor proteins within the kinetochores crawl along the kinetochore, or excuse me, the microtubule or spindle fibers. And at the very end of mitosis, those daughter chromosomes are then going to be clustered on each side of the cell. The rough endoplasmic reticulum, rough ER, is then going to help produce a brand new nuclear envelope around each cluster of daughter chromosomes. The chromosomes will uncoil to loosely expose their DNA in the form of chromatin. The mitotic spindles are going to degenerate or disintegrate to be recycled, and each brand new nucleus will begin to form their own nucleolide to upregulate ribosomal production. Shown in this figure, we can see anaphase here as a figure, and then anaphase as a photomicrograph up here. So we can see how the chromosomes have been ripped apart and are now being pulled towards opposite poles. We then have telophase here as a photomicrograph and as a figure. What I personally look for with telophase when I'm looking at a microscope slide is to find that cleavage furrow that tells me that we're now starting to divide the cytoplasm. This process of dividing the cytoplasm is referred to as cytokinesis, and that's the kind of the icing on the cake, or the cherry on top. After we've duplicated the nucleus, we need to duplicate the cell and the cell membrane and the cytoplasm itself. To achieve cytokinesis, we have a motor protein referred to as myosin that pulls on another protein called actin that will then pinch off the cell membrane, creating that cleavage furrow and ultimately giving us two separate daughter cells.
Dad, you done with your recording?
I'm almost done with this recording, Ben. Students, if you have a chance, please click on this link to watch a fantastic video that explains the stages of mitosis. And as always, if you have any questions, please feel free to post them on the class discussion board or shoot me an email. Happy studies.